Okay, hi. Yeah, thank you for the introduction, and thank you, Marisa, for this invitation. So my expertise is more basic research, so I don't really have many opportunity to interact with people from the clinic. So that's really nice for me, actually, this opportunity to really, because the idea here is trying to build this bridge between basic research and translational research. So our primary focus in our group is trying to understand the formation of human metastases in general. And I do believe that brain metastasis is an excellent model to really interrogate and understand the properties of metastatic tumor cells and to model this interaction between cancer cells and the stroma. So the data I'm going to show you today, as Michelle said, they belong to this article that we published early this year, where we apply single-cell transcriptomics to human brain metastases from different cancer types. And by applying single-cell transcriptomics, we're trying to identify commonalities across different cancer types, but also trying to understand the biology that operates in this kind of tumors. No conflicts to disclosure. So, so let's start saying some few words about some basic concept about metastasis in general. So what we know about metastasis that a cancer cell to be successful in the generation of a metastatic colony, they have to have this tumor initiating capacity. We know that the tumor cell, they develop this very complex and heterogeneous microenvironment that we call in metastasis, metastatic niche. We also know that the cancer cells interact with this niche. We are just starting to understand really what kind of biology is happening in this interaction with the niche. And also having suggested, this is mostly by using experimental mouse model, that the cancer cell, they use specific organ-specific programs that have been associated with the colonization process, right? All of this is in experimental mouse model. What we would like to know, what we don't know is, what is the nature of these programs in human metastases? What kind of form this program takes? How these programs correlate with the composition of the cell state of the stroma? And a specific question I'm really interested in my research is trying to understand why some metastatic cells, they reactivate and proliferate and progress into these over metastasis. So I put these three questions because all the analysis I'm going to show you today, they are associated with these three questions. This study started with collaboration with Dr. Joanna Philip and with my late mentor, Dr. Sina Ware. So we collected parenchymal brain metastases from different cancer types, including melanoma, breast cancer, lung cancer, ovarian cancer. And we apply single-cell transcriptomics. So today, I'm going to focus mostly in the analysis of metastatic tumor cells. I understand there is a talk later about single cells in melanoma, also applying single cell. It's going to be really exciting to see that. Today I'm going to focus mostly in the analysis in the metastatic tumor cell to identify these programs or cell state that characterize metastatic cells. And we're going to revisit these stromal clusters and the state in the context of the metastatic tumor cells. So basically trying to understand this link between these two different cell types. Okay, this is very quick. Yes, I want to say that we have different cancer types, and I want to highlight that most of the metastases that we include in this study, were located in the brain cortex. Most of the patients didn't have any treatment at the moment of surgery, but most of them, they have treatment in the past to treat the metastatic burden, systemic burden of the primary tumor. So this is something really important to say, because we always discuss this when I present this data about what is the impact of these therapies and what we see finally in the patient. It's an intrinsic noise in this when we work with human metastases. Yeah, so first we're trying to show you what kind of program the cancer cells activate in human metastasis, and then we're going to contrast this program with the stroma. And at the very end, I'm going to talk about a bit why metastatic cells they reactivate and proliferate in patients. So this is a human projection of 80,000 single cells. So what you can see here, and actually very interesting, that the metastatic tumor cells, for each one of the cases, they develop these independent clusters. And what is the pointer here? Oh, this one, sorry. So they form these independent clusters, metastatic tumor cells. We don't see any overlap between metastatic cells from any cancer, not even between the melanoma or the breast. But the stroma, we can see how the cancer cells, they cluster together. So the stromal cells, they cluster together. For example, macrophages here, T cells, or endothelial cells. If we exclude the cancer cells and just we cluster the stroma, this is how it looks, the metastatic niche in brain metastasis. It's very simple, actually. It's composed by this class of endothelial cells and mural vascular cells and immune cells. We also see a small cluster of astrocytes, but actually this was a surprise because we didn't really see that many brain resident cells in this tumor, and that is something that only when I meet with people that work in metastasis, they ask me where these cells are. So we did all sorts of staining to validate this, and we didn't really see that many, for example, oligodendrocytes or neurons that they were not detected there. Okay, so the first challenge was how we can identify common programs in this highly heterogeneous cluster of tumor cells. So to do that, we use this strategy that that is the name. Sorry, I come back here. Okay, so we use a strategy that is called NMF, non-negative matrix factorization. That basically what we did, we reduced the dimensionality of this expression file of the cancer cell per each one of the cases into a specific gene signature. In this case, we use the top 10 gene signatures that define those cancer cells for each one of the cases, and then we did a clustering based on the similarity of all of these gene lists, and that is what you are seeing here. What's interesting is that all of these 150 programs, because there are 50 patients that we include in this study, they organize in a very simple way where we see this A common or recurrent program that we call meta signature or meta programs. When we annotate this signature, we see that this signature, each one of them, they describe a specific cellular process. For example, we have two meta programs associated with proliferation, protein synthesis, response to stress, development, spliceosome activity, ECM remodeling, and epithelial to mesenchymal transition, and inflammatory response. So we have this A program that we identify that they are common in all of these cancers, including melanomas and carcinomas. What is more interesting is when we're trying to understand how these different programs correlate each other in metastasis, we observe this very simple pattern where there is two kinds of cancer cells. So there are the cancer cells that have a high score for programs associated with proliferation and spliceosome, and we have cancer cells that have a high expression for programs associated with inflammation, stress, ECM remodeling, protein synthesis, and low expression for proliferation. And we can see exactly the same here when we plot individual samples, individual cases. So this is breast cancer, lung cancer, and melanoma. And the cancer cell here, they are ordered from left to right based on the score of the program P1. And you can see, for example, like a program P3 or PA, this is protein synthesis, this is inflammation. They have a negative correlation with proliferation. And we can see the same pattern in all the cases that we study here. So what is data basically saying that the cancer cells, the metastatic tumor cells in brain metastasis, they exist in two main functional states that we call in this study archetypes. One of them is highly proliferating and high in spliceosome activity, and the other one is not proliferating but enriched in processes like inflammation, protein synthesis, ECM remodeling, EMT, and stress response. Now, if we contrast this finding with the literature, what we see is that this paper that was published in 2017, where they analyzed 868 human metastases by using bulk transcriptomics, they actually suggested that there is two kinds of metastases in patients. Metastases that they are rich in inflammatory processes, and metastases they are enriched in proliferation. Basically, the same conclusion that we see. The only difference that we show in our study is that these two states coexist in each one of the metastases. Then now we can come back to the stroma and see, well, we have these two archetypes, we have this A meta program, how they correlate with the composition of the metastatic niche. Yes, I want to show here that, for example, these are the immune cells. We see 2 clusters of macrophages, dendritic cells, 5 clusters of T cells and B cells, and all of these immune cells, they are detected in all the cases that were analyzed. Now, this analysis actually is one of my favorite analyses because it's really interesting. In this axis, we plot the expression of a specific program. In particular, we have the inflammatory program, P8, and one proliferative program here in metastatic tumor cells, and we correlate this expression in metastatic cells with the composition of the stroma in those specific samples. And then we can see how the sample that have a high score in inflammatory processes in metastatic tumor cells, they are enriched in immune cells. That is actually, that kind of makes sense if you think about it, right? Now, the question is, any kind of immune cells, or all the immune cells in general? So we went to analyze that because that is the richness of the single-cell transcriptomics, that we can go very deep into the state of the stroma. And what we saw here is it's not that any kind of immune cell. For example, we can see there's a negative correlation in the expression of inflammatory processes in metastatic tumor cells with, for example, macrophages ApoE positive. But what is more interesting, even in the T cells, is different. So they have a positive correlation with T cell clusters that they are enriched in processes like a T cell activation or cytotoxic score, and they have a negative correlation with T cell clusters that are enriched in T cell energy or suppressive programs. So it's the flavor of the immune cells actually is correlated with the flavor of the cancer cell they're going to take into in this metastasis. So the communication between these two different cell types is really important to really understand what biology operates in this kind of tumor. Then basically, this is the same conclusion, just using a different approach. This is a principal component analysis of the metastatic tumor cell, showing basically the same conclusion that we had with the NMF, that there is 2 kind of major programs that drive all the heterogeneity of the cancer cell, that is inflammation and proliferation. So we really wanted to understand how this happens. So can we really understand a bit the biology, how the cells progress from quiescence, not proliferating, into a proliferative state? And that's have a very big implication to understand why metastases, they actually reactivate in patients. We know that the cancer cells, they can be dormant for many years in some cases, and then just suddenly reactivate. So we really wanted to do that. So what we did here was we scored the cancer cell per each one of the cases into cycling metastatic cell versus non-cycling metastatic cell. We ran a differential expression analysis between these 2 states, and then we identify the intersect of the common genes that they are differentially expressed between these 2 different states in all the cases analyzed. And that is what you are seeing here. What's interesting is in the top 10 genes of the in the top 10 of the gene that the metastatic tumor cell, they have a high proliferation score, they are downregulated genes associated with antigen presenting genes. So basically, MSC class 1, they are all of the genes associated with that system, they are downregulated in cancer cell, they are being highly reactivated. What we propose here, what we believe that what we have here is a specific state where the expression of this gene and the downregulation of the gene, they define the escape from quiescence and the reactivation in patients. We don't have functional validation. We are trying to really understand what is the role particularly of this gene, for example. We know that many of them, they have a negative, they are negatively correlated with prognosis in patients, but we don't know really what kind of biology is happening here. So it's really interesting trying to understand this, and especially how this correlate with the idea of escape from the immune attack. So in line with that idea, so this is the same paper that I showed before. We correlated the expression of these genes with the composition of the immune fraction in 868 human metastases. This includes brain metastases, but also any kind of other distant metastases. And we can see that those genes that are being down-regulated by cancer cells, when they reactivate, they correlate with the increase of immune fraction. Basically in line with what we propose here, that the reactivation from quiescence and immune evasion, they can be two events, they can be connected. I know it's very provocative, that idea, but we believe that there is something that we are not seeing, how the cancer cell actually they can escape. Another thing to say here that I didn't show this data just because the time is very short to show this, is that all of these metastatic tumor cells, they are likely clonal. We don't have exome sequencing, but we did copy number variation analysis in these metastases, and we saw these highly homogeneous profiles in metastatic tumor cells. So all of these are happening in a non-genetic way. So these two archetypes, there is some level of epigenetic maybe that is driving all of these programs and different that we see here. So we validate this idea doing some immunostaining. This is a collaboration with a group in Sweden, basically showing the same, that the proliferation of cancer cells and the infiltration of immune cells have a direct correlation. Okay, so today I show you that this idea, what kind of program the cancer cells actually activate in human metastasis. We identify a different program. This A program defines two functional states that we call in this study archetypes, one inflammatory and one proliferative. What is interesting is this non-proliferating state. The quiescent one is very dynamic, looks like. The increase of protein synthesis, ECM remodeling, EMT, is very intriguing and very interesting to really understand what these cells are doing when they cannot proliferate. We also show that the context matters, so the interaction with the stroma play a major role in these states that the cancer cell acquire. And we also suggest that the proliferation, the reactivation and proliferation of cancer cells could be associated with the idea of immune escape. I'd like to finish saying some few words, just 2 slides, about what is next, right? Because I think it's important to show where this is going. In this moment, we are really trying to understand what molecular determinants they regulate these archetypes. So we are doing, can I come back here? Here. Chromatin profiling to identify which transcription factor or transcriptional regulator they can be associated with these two different states and trying to connect these different programs. And more interesting, and that is because I really like the people that we work in basic research. We are trying to always develop new systems, trying to see how we can develop new ways to model this interaction between cancer cells and stroma. And I want to show you 2 slides about what we are doing in our lab now with this technology that is called tumor organoid technology. This, for example, is a brain metastasis from lung cancer patient. And in green and in red, we have organoid-specific CD8 T cells that were primed with the same organoids. And I want to just show you very quick this video showing after 72 hours, the cancer cell, they induce a very nice immune attack against the metastasis in 3D. Not all the cancer cells die, as you can see. So now we are trying to really understand this cell that can escape from the killing, what kind of program, and then trying to contrast this finding with the human metastasis. Right? This is trying to model this tumor immune interface. But also, we are trying to model the tumor stroma, not just immune, but using the combination of these iPSC-derived brain cortical organoids in combination with human brain metastasis. We are actually really doing this. This is one of the first pilots that we have been doing, but also now we can even reconstitute this organoid that they have different kind of cell types that they are present in the cortex, for example, astroglia and all kinds of neural progenitors and mature neurons. We can add microglia, even immune cells, and then we can put these 2 systems together, and then we can see how these cancer cells, they infiltrate the organoid, the cortical organoid, and they can actually really grow there. It's a very nice system to model what is happening with the cancer cell when they are in contact with all the brain resident cells and they're trying to expand. It's a very nice system that we are very optimistic what kind of biology we can obtain for this system. Well, many people involved in this effort. Well, I just to mostly focus in my late mentor, Sina Ware. She was the person who really encouraged to go into all of this, and she saw the potential to study brain metastases, but not with the real interest of understanding brain metastases, like a really nice model to understand human metastases in general. And Joanna, so we have been working with Joanna for many years, and also Marisa. So yeah, thank you.